Okay, so this one, I've only got a couple left, is movement across membranes. And really this is just to summarise what you've just been doing. So we have 3 types of movement across membranes. Diffusion, and here I've drawn more particles and fewer particles. So diffusion is the movement of particles from an area of high concentration to an area of low concentration until equilibrium. We say that the net movement, that means overall, is in that direction from high concentration to low concentration. And that's because particles are moving in all directions. However, the majority, overall, the movement would be from high to low. This is things like gases in the air, in the lungs, gas exchange. Lots of diffusion happens all over the place. Osmosis is a special type of diffusion, and again it's along a concentration gradient. So here, from high to low, we say that's a concentration gradient. However, with osmosis, it's specifically and only limited to water moving across a partially permeable membrane. And permeable means it can let things through, partially means it can only let a few things through, and it's based on size. So if there's no partially permeable membrane and it's not water, then it's not osmosis. So here's our partially permeable membrane. Here is our water, because we're talking about osmosis. Water can fit through the partially permeable membrane. We've got a gradient here. Now it's not really a concentration gradient, because concentration is usually to do with salts and dissolved substances. But the concentration gradient here is actually, we say it's a water potential gradient. In other words, it's a high amount of water relative to solutes here, and it's a low amount of water relative to solutes here. The final one is active transport. So active transport, you can see I've drawn a little membrane here and some weird little sausage thing. And this is what we call a carrier protein that we have in membranes. And you can see that we're going in the opposite. We're not doing from high to low, like going down a slide. We're doing from low to high here. And if you imagine trying to climb up a hill or climb up a slide, it's hard work, it takes energy. So we've got low concentration of solutes, of whatever it is, salts. We've got high concentration. And in order to get from a low to a high concentration, the particles need to be pumped across. And this requires a physical pumping, and it uses energy. It's actually something called ATP. But that's our little package of energy that is used up in order to push it across. And this happens particularly in the roots of plants in order to get minerals across. Active transport. Now, you may have done an experiment, which was your osmosis in potatoes. And I've drawn a potato here. So here's a chip. And this is our starting chip. And you should have measured the dimensions. And you could have worked out the volume. Or alternatively, if we'd done this in class, I would have weighed it. We worked out the mass in grams. Some of those chips got bigger, some had no change, some got smaller. And the ones that get bigger are generally the ones that you put into only water or a very, very weak solution of, say, salt or sugar. The ones that have no change, there'll be a very specific concentration of salt or sugar in water, and this will be the point at which the salts inside the cells of the potato are at the same concentration as the salts in which they were put. So the beaker that you put your salt solution in is at the same concentration that the salts are within the cells in those potatoes. They get smaller, they shrink, if you have really, really salty water or really, really sugary water. And this is because of osmosis. So in this one, the water is exiting the chip, trying to balance out the water in that really salty solution to make an equilibrium. In this one, the water is moving in through osmosis into the cells of the chip, because this is much more concentrated in terms of salts and sugars inside the chip than the water itself. So that's the results that you hopefully would have got. And there may be one where you get no movement whatsoever. And finally, this graph to some of you will look really familiar. And you had to work out the point at which is the isotonic point at which there was no movement in or out. And if you didn't get a chip that didn't change at all, we can work it out from a graph. So in order, you have to know how to do the percentage change in something. So in this case, my example is in mass. And that's really, you work out the change in mass. In other words, what was it at the start, what was it at the end? And the original, the starting mass, and you times it by 100 and you get a percentage. And in this experiment, you're going to have some that grew, so that's a positive change, percentage change in mass of potato chip. You're going to have some that shrunk. So typical results might be that when you've got lots of, so this is the concentration of salt, when you've got lots of water, you're going to have a big gain in mass. You're going to have a slightly smaller gain in mass as your solution becomes less concentrated. Let's say we had something like here, and as we get more concentrated, we're going to find that we'll have the chip is shrinking. So you might end up with a graph that looks something like this. Some of your graphs did look like that. And we can see from here that we can work out the concentration. I've just made these numbers up, but we could work out the specific concentration of the liquid inside the cells of that potato, and that would be the point at which it crosses the zero line, where there is no gain and no loss in mass. So that's our equal point, the isotonic point. And that's it. I think that's it. Good luck with your revision.